Good morning, everyone. I'm honored to be here to present to you our latest work that focused on the perceived emotional experience during a naturalistic stimulation. And we focused on the emotional experience since emotions are pervasively present in our everyday life, and they can be elicited not only when we experience something in first person, but also when we simply observe other people interacting. Emotions can be characterized along several dimensions, such the polarity, so the valence, and the intensity, the so-called arousal of the experience. But since emotions are multifaceted and complex, other states can also arise, such as ambivalence, which is the presence at the same moment of positive and negative emotions. So the question arises itself, can we actually represent and study the complexity of emotions? And one possible good way to do so is by using movies in our research, since movies can easily lead to an emotional contagion between the observer and the character state. And that is the reason why we chose to employ a naturalistic stimuli, so a movie, in our study, to assess the perceived emotional experience and to answer some questions that were, so first of all, whether the emotions elicited by a movie could explain the brain activity of an independent group of subjects, and if so, which brain regions are encoding the emotion dimensions and whether they are represented in a distributed way, and lastly, which features of the dimensions are encoded in each region. So to answer these questions, we started from a high-quality publicly available dataset that you can find at the studyforest.org that comprehend data from a group of subjects that, while lying in the fMRI scanner, watched an edited version of the movie Forrest Gump that lasted for approximately 2 hours. So this is the single subject preprocessing pipeline. For the anatomical data we used ANS and AFNI, and for the functional data we employed AFNI. And as you can see, we thoroughly preprocessed data, however keeping the number of interpolation steps and minimum. And so at the end of the preprocessing we obtained for each subject a preprocessed TPI that then we averaged together, obtaining a group level time series, and from this time series we regressed out the time series of the low level acoustic and visual properties of the movie. And after that we temporally smoothed using a 10 seconds window, obtaining a group level time series that constituted the dependent variable in our analysis. So now we're missing the emotion part, and to obtain that we asked a group of subjects to watch the exact same movie that was employed in the fMRI experiment and to give a continuous rating of the perceived intensity of 6 basic emotions that were happiness, surprise, sadness, anger, fear, and disgust. So we obtained 6 regressors, one for each emotion throughout the movie, that then we concatenated that downsample to match the fMRI temporal resolution. And then we also looked at the agreement across subjects in the emotional rating, and it was consistent, as you can see in the dissimilarity matrix, having an average correlation of approximately 0.4 on a total of 3,600 time points. And here it represented the group level time series and the moments in the movie that correspond to the peak of intensity in the rating for each emotion. So following the exact procedure that we used for the fMRI data, we averaged single subject regressors. We temporally smoothed using a 10 seconds window, and then on the pre-processed group level regressors, we computed a principal component analysis that highlighted the presence of several emotion dimensions, such as valence and arousal, and the second PC is what we interpreted as ambivalence, since happiness and sadness have the same load on this component. And together, the first 3 components explain more than the 80% of the variance. Altogether, the 6 principal components constituted independent predictors in our analysis. So our full model was made of the 6 PCs as regressors, and that is what we fitted in the brain activity using a voxel-wise encoding, obtaining a full model R-square map of this fitting. And to assess the statistical significance, we used a phase shuffling method with 10,000 permutations, obtaining 10,000 null models that then we fitted again in the brain activity, obtaining 10,000 R-square map of this fitting that constituted a null distribution on which we computed the p-value. value. And to overcome the multiple comparisons problem, we employed a false discovery rate correction that finally led us to the map of significant results. So these are the results for the full model, answering the question which brain regions encode the emotion dimensions. And as you can see, we can observe regions that we know already being involved in emotional processing and social cognition in general, such as CPJ, posterior SCS, the inferior frontal gyrus, and supramarginal gyrus. And also we can observe a dominance of the right hemisphere compared to the left one, also in line with previous findings. After that, we wonder whether the emotion dimensions are represented in a distributed fashion, and to do so, we looked at the relative contribution of each dimension, and here we color-coded them, so valence corresponds to red, arousal to blue, and ambivalence to green, and the luminance of the color also corresponds to statistical significance, so a brighter color means a smaller p-value that we sampled from a log distribution. So as you can see, we can observe regions in which there is a dominance of a single dimension, such as in the bilateral posterior MTG for valence and in the medial superior frontal gyrus for arousal. But what is actually more interesting is that we can observe in some regions the presence of a gradient. So in posterior STS and TPJ, we can see that the more inferior territories are actually encoding valence, while the more superior ones are encoding arousal. And also in the precentral sulcus, we can see that the posterior part is encoding ambivalence, while the anterior one arousal, a mixture of arousal and valence. After that, so we wonder which features of the dimensions are encoded in each region, and this is the map for valence. So here, the blue color means that the higher the bold is in that region, the more positive was the valence associated to that scene. And we can observe most of the regions having a behavior like that. Just in the rostral middle frontal gyrus, we can see that in this region, the higher the bold is, the more negative is the valence. But again, when we look at TPJ in posterior SCS, we can observe a gradient. So in more inferior territories, the higher the bold is, the more positive is the valence, while in more superior ones, the higher the bold is, the more negative is the valence associated to the scene in the movie. This is the map for ambivalence, so we can observe peaks of activity in interesting regions, such as TPJ in the inferior frontal gyrus and supramarginal gyrus. But when we look at the precentral sulcus, we can see that only the posterior part shows a positive correlation with the bold signal, while the anterior one does not, in line with what we previously observed in the emotion dimensions gradient. Lastly, this is the map for arousal. So here also we can observe interesting peaks of activity in TPJ in the inferior frontal gyrus that are in line with previous observations. So in conclusion, in our study, using a naturalistic stimuli and a continuous emotional rating, we could observe a dominance of the right hemisphere compared to the left one, and particularly in some regions. And when we look in these regions, so TPJ, posterior SCS, and the precentral sulcus, we can see that these regions are encoding the emotion dimensions in a gradient behavior-like, so in a distributed way, while other regions are showing a dominance of a single dimension. And this gradient is also observed when we look at the features of the emotion dimensions. So TPJ and posterior SCS show an inferior to superior gradient, where the most inferior part, the higher the bold is, the more positive the valence, while in the superior one, the higher the bold is, the more negative is the valence. And while in most of the regions, there is a behavior like the higher the bold is, the more positive is the valence. Just in the right rostral middle frontal gyrus, we can see that the higher the bold is, the more negative is the valence. So for this last slide, let me continue being emotional. And first of all, I want to thank the people behind the Study Forest project, because without them, we wouldn't be able to perform the study. And my lab, the MOMI lab at IMT Lucca, and Luca Cecchetti, who I'm very lucky to call advisor. Thank you.